There's a few examples where there's a deletion or a duplication, but right now I think like 9 out of 10 of the mutations that I've seen are just a single letter of DNA. And that single letter can affect things in different ways. So one of them is what's called a missense mutation. So you see up top here, these 3 letters, CAT, those are the DNA letters, that encodes for a single amino acid that's called histidine, that's represented as HIS here. And then underneath, what they're showing is that that CAT has changed, so that little letter in the middle has changed from an A to a C. And instead of coding for histidine, it now codes for proline. Okay, and you might, get the little cursor back here. You might be saying to yourself, well, what's the big deal about that? It's just one amino acid. The analogy I gave yesterday was trying to, if you imagine that DDX3X is a machine, like a machine that, say, assembles cars in a factory, and it's a robotic machine, meaning that it's not, there's no human operating it. It has to do the same thing over and over with incredible precision. And so that histidine to proline, so proline, histidine is an amino acid that's kind of flat and has a little thing sticking out like a lollipop at the end. And proline is like a severe right angle in the structure of the protein. So those are very different from each other. And so a giant right angle is going to cause that protein to twist in unexpected ways and probably affect its function. So that's how, even though it's a tiny change, it can have large consequences. Okay, another kind of mutation is called a nonsense mutation. Another way of referring to that is a stop codon. So what that means is that you're going along, there's something called a ribosome, which is reading the RNA and making that into the protein. So that's, again, another machine inside the body. And it's making it, it's going along reading 3 letters of DNA at a time. And then it comes along, and instead of seeing up here CAG, which would be glutamine, it sees TAG, and that's a stop codon. So that's a signal to that ribosome, to that protein-making machinery, to stop. It's saying, oh, I'm done making the protein, and what happens is the ribosome dissociates from the protein, and the protein, in theory, should now be able to go to where it's supposed to go to do its function. But what happens when there's what's called a premature truncation, so meaning that that stop codon shows up in the middle of the protein or even in the beginning of the protein, that there's a self-editing function in the cell that says, oh, that protein looks wrong, and it actually chews up that protein and gets rid of it. So in girls that have a nonsense mutation, you're going to likely have one X chromosome making the normal protein and the other X chromosome not able to make anything. And so that leads to not enough DDX3X, so 50% of what you'd expect, and that results in the condition. Okay, and there are 2 others. One is called a frameshift mutation. So again, there's a lot of high school biology here, and I apologize, but it's important. Again, it's the 3 letters at a time that get read from DNA to RNA and then RNA to protein. And again, here, if you have in this case, the example given is an insertion. So an A gets inserted, so it's CAT, CAT in the top, and it's CAT, and then there's an A in front of the next C. So what that does is it tricks the ribosome, and the ribosome then reads it as CAT ACA, which is, instead of what it's supposed to be, which is histidine, it's threonine. And then it goes along making the wrong letters because it's now out of sync. And it keeps reading those wrong letters typically until it gets to a stop codon, which is usually what ends up happening. And then again, those proteins tend to be degraded because they're not functioning normally. And then the last kind, there's some other examples as well, but I'm sticking to ones where it's just a single letter. The last kind is what's called a splicing mutation. So again, and I apologize, this is like, all of biology 101 in 10 minutes. What happens is the cell makes a copy of the DNA into something called RNA, and that RNA is what's read to make the protein. But the RNA actually needs to be chopped up into little pieces and put together so that it can be read in the right way to make the protein. And to do that, you need splicing to occur. So the RNA gets made in this really long fragment, and then it gets spliced down to just the right size to be read to make a protein. And there are errors of that splicing. So at the beginning of a splice, you typically see these two letters, and at the end of the splice, you see these two letters, so AG and GT. And the splicing machinery, again, there's a machine inside of the cell that reads that, and then it splices it together appropriately. So you have part 1, part 2, and part 3, they're called exons, and then they get fused to form 1, 2, 3, and that's what you're supposed to see. And then it can be, with a mutation, it can be incorrectly spliced. You can have 1 spliced to 3, or you can have just 2 spliced to 3, and you can have other errors as well. Typically what this means is you have a reduction in the amount of normal DDX3X that's being made. Okay, so you can have missense mutations that tweak the machinery, or you can have other mutations that mostly lead to less of the protein being made. Okay. All right, so then what Luigi did is he made a map of, and again, this is probably not completely up to date, of, at the time when he made this, all the mutations that we had. And I think we're going to post all of these slides, right? So everyone will have access to them, so you don't have to worry about it. But what I want to point out is that there's two, so DDX3X, like all these other things that I've been talking about, is a machine that does things. And one of the things it does is it seems to unwind RNA to make RNA accessible to be translated. And so it's a machine as well, and there's kind of like the main well understood part of the protein that's actually the machine, as opposed to, say, the part that allows it to attach to different parts of the cell. And that's this blue part here, this bluish purple part here, and here is kind of the main what we call the helicase part of the protein. And you can see that most of the mutations, like 90% of them, are inside of either this first ATP binding domain or this second C-terminal helicase domain. And so that tells us that that's why your daughters have developmental issues, because these mutations are occurring in parts of this protein that are sensitive to particular changes.